Welcome to the second webinar from Protein Stable. Antibody engineering with SUPR-CM. I'll look at the SUPR-CM workflow and how the system can enhance the antibody engineering process by Dr. Alastair Davey. Now, this webinar is going to be about 20 minutes long, and then there'll be about 10 minutes at the end for questions. Please put your questions into the chat box, and then we can answer them at the end of the webinar. If we can't get to your question within the allotted time slot, we will send an email directly to you with an answer within the next day or two. So we will reply to all the questions. Okay, so I'm Lindsay Cole, the Research Director and the host for the day from Protein Stable. And then we have Alastair Davey, who'll be presenting today. So I think that is all we have to do. So I'm going to hand over to Alastair, who will now take you through antibody engineering with the SUPR-CM.
All right, thank you very much for that introduction, Lindsay. And yes, hello and welcome everyone to this Protein Stable webinar. My name is Alastair Davey, and today we're going to be taking a look at our new product, the SUPR-CM, and how you can integrate it within an antibody engineering program. So we'll start off by looking at a recent publication where they utilize chemical denaturation as an orthogonal technique to use as part of an antibody screening program. And then we'll ask the question of what the SUPR-CM can do to improve that type of process, what it brings to the table, and what we can do to give you confidence so that you can introduce chemical denaturation into your own antibody screening programs. And we'll show you some data we've generated with the SUPR-CM data to help explain the advantages and show off the capabilities of the SUPR-CM. So let's start off by taking a look at chemical denaturation and how it's been utilized in the field as part of an antibody screening program. So for the purposes of illustrating chemical denaturation's usefulness within this type of application, we're going to be referring to a recent publication by Kerwin et al. The citation's there at the bottom of the slide in case you're interested in looking that up. Now in this particular study, they were looking at different mutant strains of a parent antibody, a broadly neutralizing antibody, that they wanted to use and develop as a prophylactic tool against the spread of HIV. Now, understandably, since HIV is the target virus, then the target market for this particular product is going to be sub-Saharan Africa. And in this context, the stability of the antibody became quite important since the cold chain is not always guaranteed. Now, what we mean by the cold chain is we're talking about the refrigeration that the antibody that these products need, not only in terms of logistics, transportation, but also in terms of storage of these products before they're used to treat the patients. And in this region, in this market, particularly rural areas, it's not always guaranteed that you're going to have refrigeration capabilities. So you need to ensure that your antibodies are stable enough so that they can survive the 40-degree African sunshine and still function reliably and be able to do their jobs correctly. So in order to assess different variants of the parent antibody, they assess the stability through 2 rounds of measurements. So in the first round, they looked at single variants of the parent antibody. So this could have been a change to the light chain of the antibody, or it could be a change to the heavy chain of the antibody. And they were assessing the change in stability of these antibodies through techniques like thermal denaturation via differential scanning fluorimetry, or there was a low pH aggregation study that used high performance size exclusion chromatography, and then there was a neutralization assay that was there to ensure the antibodies were still doing their job, that they haven't lost any functionality of the antibody. And so after the first round of measurements, they are able to identify a few select single variants that increase the stability of the antibody without compromising on the functionality. Then in the second round of screening, they generated a new list of constructs where they combined the different variants together so that they could further increase the stability of the antibody. And it's in this round that they introduced chemical denaturation as an orthogonal technique to assess the stability of antibodies. And from that data set, they were able to then decide upon a final list of candidates to move into the last stage of screening and finally come to their decision as to what the most optimal candidate was. So we're going to take a look at the second round screening data, and I've cropped this graph from the paper, and I have to give my hats off to the authors. They've managed to cram a lot of information into this graph. I'll do my best to explain each aspect of it. So this scatter plot combines the differential scanning fluorimetry data, the size exclusion data, and the chemical denaturation data into the one plot. So looking at the y-axis of the scatter plot, we have the thermal denaturation data. Now, interesting to note that the TM values weren't enough to distinguish the two populations of antibody variants. They had to go in and do further calculation to generate this weighted shoulder score, which essentially boiled down to a sort of scaling factor that helped emphasize the changes between the first and second TM values. And then on the x-axis of this graph, we have the chemical denaturation information. So we're looking at the unfolding inflection points. So this is the concentration of denaturant that's needed to get half the antibodies into the unfolded state. No further calculation was required. The information was enough to distinguish the two populations of antibody variants. Then we have a color bar on the right of the plot, and this is where the size exclusion data comes into it, looking at the percentage of high molecular weight species. And then finally, we're looking at the shape of the scatter points. So if it's a circle, we're looking at oligomer percentages below 2%, and if it's a star, then the oligomer percentage is above 2%. And the lines emanating from each scatter point congregate at the mean value for that population. And this is the main takeaway that's gained from this data. We have two distinct populations, two groupings of antibody variants. And when the authors looked into what variants were behind these antibodies, they were able to see that one of these populations had a particular variant, whereas the other population didn't have this variant, this being the HVT108R variant. And so from this data, including chemical denaturation, along with the thermal and the size exclusion data, they were able to identify this crucial variant that helped increase the stability of the antibody. And so with this data, they had a good grouping of antibodies they could look at. Then assessing the neutralization data, they were able to whittle the list down to 3 main candidates that they could push on for the final screening round, this being MS200, MS201, and MS202. And so in the final round, they looked at a small-scale purification study, simulated viral inactivation, accelerated stability, a PK analysis, and polyreactivity analysis. And so here I've extracted the table 3 from the publication, which contains the accelerated stability study here. This is where they incubated the antibodies for 6 and 13 weeks at 40 degrees. And if you look at the numbers, you can see that MS202 has performed the best there. And it was this particular antibody variant, MS202, that the authors concluded was their most optimized form, and that would be their ideal candidate to progress with the rest of the development program. And so we've been able to show and be able to see with this publication that chemical denaturation can be a useful tool and can provide orthogonal data to help make decisions about which candidates to progress with when you're doing an antibody engineering program. Now, there are certain things that might limit the use of chemical denaturation within this application, and we've identified those limitations or these challenges as low quality of data, low throughput, slow acquisition time, particularly if you're using a conventional microplate. Then it's got a monochromator based system, so you're having to scan through all the wavelengths to generate a full fluorescence spectrum. And that can then have consequences on the throughput capabilities of your measurement. And then finally, there's a challenge with the needs for high sample usage, and that might limit the use of chemical denaturation within this type of application. And so, having identified these challenges, the question then becomes, how can the SUPR-CM improve this type of application? What advantages does it bring to the table that means that you can use chemical denaturation within your antibody screening program? And so let's start by introducing the instrument itself. So this is the this is the SUPR-CM instrument, our brand new instrument from Protein Stable. It is a intrinsic fluorescence plate reader that works with 96 and 384 well microplates, a convenient format that helps reduce errors, increases throughput, whilst reducing consumable costs. The SUPR-CM is capable of performing full spectrum measurements, as you can see on the plot on the right, and we're doing chemical denaturation experiments. So this is where we have the antibody in a titration of different denaturant concentrations, and we measure the change in fluorescence spectrum with denaturant concentration. So comparing the 0 molar case and the 6 molar case here, we can see there's a clear shift in the fluorescence spectrum of the antibody. And so we can quantify that change. Commonly, what parameters used is the ratio of intensities, which we've got shown on the plot on the right here, this showing our denaturation curve. And we've got our characteristic features here. We've got the lower plateau region, we've got the upper plateau region, this being our folded and unfolded states respectively. We have the transition region highlighted in the shaded area there. And then we have our CM value, that's the same as the inflection points that I showed in the scatter plot from the publication. And so with the SUPR-CM, you're able to generate this type of data, and importantly, we're able to get this data fast. So here we have an example of an IgG1 monoclonal antibody being measured. So we have our denaturant concentrations going from 0 to 6 mole from the left to the right, going across the rows. We've got this in triplicate as well, and you can see that the SUPR-CM is able to measure this data very, very quickly, generating full fluorescence spectrums here, and whilst the measurement's progressing, we're calculating our ratio of intensity values, which we then use to generate our denaturation curve there. So there we go, we were able to illustrate the rapid acquisition of data with the SUPR-CM. So now I'm going to move on to look at some of the data we've generated with the SUPR-CM. And for the purposes of illustration, we're going to look at a force degradation study. And the reason I chose this study was because it's a good analog. It emulates the type of changes you can see when looking at different mutant strains of a parent antibody, like in the publication I talked about earlier on. So to start off, we're looking at a deamidation study here. This is where the antibody solution was incubated for 1 week at 40 degrees Celsius. And we can see that there's been a significant change in the stability of the antibody after this incubation period. Looking at the denaturation curves on the left, we can see that after incubation, the transition regions have shifted towards lower denaturant concentrations, signifying a reduction in the stability of the antibody, since less denaturant was needed to unfold the antibody. And you can see from the data as well that we've got 2 distinct transition regions there, signifying that this antibody has a 3-state behavior to it. And that is something the SUPR-CM is able to pick up on, signifying the quality of data that you're generating. And with the SUPR-CM, we can then fit a three-state function to the data and generate our Gibbs free energy values and our CM values, which allows us to quantify the stability of the antibody. And comparing the CM values for the different transition regions, it tells the same story. We're seeing a reduction in the CM values, signifying that the deamidation has lowered the stability of the antibody. Next, we've got a glycation example here. So in this case, the antibody was introduced to a glucose solution and then incubated for 1 day and 7 days. And then we compare the different examples together. So again, the glycation process has reduced the stability of the antibody. The transition regions are shifted towards lower denaturant concentrations. The CM values back this up. We can see there's a decrease in the CM values after glycation. Interesting to note that there's not much change between the 1 day and the 7 day glycation example. So we can say from this that the glycation process was shortly over after 1 day. And so the SUPR-CM can be used to monitor stability of antibodies throughout a process or over a period of time. Then we have a peroxide degradation example here. So we introduce the antibody to a peroxide solution, let that incubate for a few hours, and we're comparing it with a control sample. And this is quite an interesting story here. We see that we've still got our three-state behavior, but only one of the transition regions has been affected by the introduction of hydrogen peroxide. And this is confirmed when we look at the fitted values from our three-state model. The CM values for the first transition region, this is CM1 I'm looking at. We can see there's a significant change there, a 0.2 molar change in the CM value. Whereas if we look at the second transition region, we don't see the same change in the CM value. It's roughly 0.03 molar change there. And so this illustrates how the SUPR-CM is able to measure subtle changes in the antibody stability. For instance, if these two transition regions were related to different domains of the antibody, then you'd be able to see a change in stability of one domain as opposed to another domain. And that would be quite powerful information to have as you're performing your antibody engineering program. And then finally, the last bit of data I'd like to show you today is an example where we use a very low amount of antibody. So we've got the same sort of experiment being performed with 24 denaturant concentrations, but in this case, each well only has 300 nanograms of antibody. And we're still able to get good consistency in our data. We can still identify our three-state behavior, and three-state model converged on the data set as well, so we could quantify the stability of the antibody. And so bringing the presentation round and introducing the challenges I talked about and now we can see how the SUPR-CM overcomes these challenges. We've been able to show the SUPR-CM can acquire chemical denaturation data of a high quality and with high detail, so you can really dig into the minutiae of the antibody stability and ensure you're making the best decisions about which constructs to move forward with. We can work with a 96 and 384 well microplate, which can help improve and increase your throughput capabilities. And it's also a convenient platform that helps reduce costs as well. We can scan a plate very rapidly, as we've shown. If you can have a 384 well microplate with, say, 5 different antibody constructs measured in triplicate, with 24 denaturant concentrations, and that would fit on the one 384 well microplate, and you could then acquire denaturant and stability information in about 2.5 minutes. And then looking at the low amount of sample that's needed, we looked at the publication I've talked about earlier in the presentation and compared the amounts they were using with the 300 nanogram example I showed in the last slide. And we calculate that if the authors had used a SUPR-CM instrument, they could use 30 times less antibody and still get the good quality chemical denaturation information. And so looking at the time, I think it's about time I wrap this presentation up. The only thing left for me to do is to thank all of you for joining us and for listening to me present. I think we've got some time for questions, so I'll pass the ball back over to my colleague. Thank you very much.
Okay, thank you. Now, if you can put your questions in the chat box, we'll be able to answer them. Alternatively, if you don't want to answer it here or have a question later, you can obviously email us at the email address up there. Okay, a question, Alastair. Are the measurements done in sequence for each well or simultaneously on the whole plate?
Good question. So the SUPR-CM utilizes an array detector, and each well is measured sequentially. But you've been able to see with the video I showed earlier on, you could see the sequential measurement of each well as the plate was being scanned across the well. So yes, the SUPR-CM measures each well sequentially.
Okay, thank you for that. All right, I will. I guess the other one is, how are you actually evaluating the fluorescence signal?
Right, okay, so in this case, we're looking at the ratio of intensities. So you'll remember the plot of fluorescence spectra I showed, where we had the 0 molar and the 6 molar case, and you saw there was a clear shift in the spectrum there. So taking the peak values of those spectra, we can then go through and calculate the ratio of intensities. So commonly, the ratio of intensities use the wavelengths 350 nanometers and 330 nanometers. And you can calculate a simple ratio there, plot that against denaturant concentration to generate your denaturation curve. The SUPR-CM software also allows you to look at just the fluorescence intensity at a particular wavelength, and you can also look at the barocentric mean value as well. So you've got a few options to play about with if ratio of intensity isn't your favorite method.
Okay, I guess one more question, and I think unless somebody else has any, how does this compare to a standard plate reader for this assay?
Yeah, so it's a good question. Comparisons are always interesting. So most conventional plate readers, they utilize a monochromator-based system. So in order to generate a full spectrum, they have to scan through every single wavelength in order to generate that type of data. So that obviously takes some time. Even if you're using a very low integration time, it still takes a substantial bit of time to measure a full spectrum. And then if you multiply that by, say, 384 wells, it then starts to stack up. And so data acquisition times and throughput can be a challenge when working with conventional microplate readers. And the SUPR-CM, as you've seen, we have very, very good quality data. So then that's another thing to consider as well, is the quality of data being produced. Because if you're trying to reduce the acquisition times or increase your throughput, then you're having to compromise on the quality of data. But with the SUPR-CM, you don't need to compromise. You can measure quickly and still get good quality data.
Okay, thank you for that, Alastair. If there is no more questions, I think we'll end the webinar here. Now, just be aware, we have recorded this webinar, and we will be sending you a link to a copy of this for your own reference and to hand on to colleagues and other people. Thank you for your time and thank you for attending. If you have any other questions, the email address for Sales of Protein Stable is the place to go, and we will be more than happy to answer any of your questions going forward. Thank you for your time and have a good day.
Thank you very much, everyone. Take care.